This screencast is about monohybrid crosses. Before we can go into talking about a monohybrid cross, we need to do a little bit of review from the last chapter. We know that when it is time for successful sexual reproduction to occur, there will be the fertilization of sperm and egg. Inside of sperm and egg are 23 pieces of human genetic material. And when the 1N or haploid egg and the 1N or haploid sperm combine, we know that we will make a zygote that will contain all 46 pieces. So we have 23 pieces here, shown in blue, representing dad, 23 pieces here, shown in red, representing mom. When fertilization occurs, the 2N number will be 46. And for every single genetic pair group, there will be a matching one from mom and a matching one from dad. Located on the surface of these genetic pieces are regions that represent the areas for a gene. The official term for a region that represents the area for a gene is called a locus. And so if we wanted to talk about handedness being the gene, and we wanted to recognize the locus being on, say, maybe this pair right here, and if we're talking about the fact that every single person gets one form of the gene called an allele, one allele from mom. Here it's represented as a capital letter G. Here is the representative allele or form of the gene from dad that can be found on the blue piece of genetic material that was given in the sperm. And we can see that the dad gave a lowercase g. We'll talk about what the upper and lowercase represent in a while. So anyway, a loci is a region found on genetic pieces. So let's say we wanted to talk about whether or not you can roll your tongue. And let's say that that would be found on that chromosome pair of pieces, one from mom and one from dad. And so you would then look onto those genetic pieces and you would be able to find the locus or the area for the, say tongue rolling, whether or not you could be able to tongue roll. And your mom will give you a version of an allele, your dad will give you a version of an allele, and together they will work together to give you your specific trait. Now remember, you get 23 pieces of genetic material from the mom in the egg. You get 23 pieces of genetic material from the dad. All of these 46 pieces have matching pairs. Here we can see one matching pair. But remember that when this cell begins its cell cycle, it will go through the S phase. And each one of its red pieces will be doubled, eventually forming into a chromosome during prophase. So here, if we're looking at the red piece that originally came from the egg, this is what the red pieces would look like after S phase and during prophase when they coil into a chromosome. So remember that you get chromatin from your mom and your dad from sperm and egg that will eventually form into chromosomes during the cell cycle during prophase. So that's what this diagram is wanting to show you, that when you form the chromosome from this red piece, they will both be carrying the exact same alleles from your mom. And when you make the copy of the chromatid for this piece, they will also carry the exact same copy of the allele from dad. So recap, sperm and egg create diploid zygote. Inside of diploid zygote are 23 pairs of chromatin. On the matching pairs of chromatin are regions of genes called loci or locus. Locus is one. Loci, here I can see 4 loci, 4 different genes. And so individuals are given 2 alleles when sperm and egg unite for every single gene in your body. So now what we want to do is we want to talk about Mendel's experiments and why he kept on coming up with this ratio over and over and over again of 3 to 1. So basically what would happen with Mendel is when he took a parental generation and he crossed a dominant and recessive, and let's do purple and white. If he crossed a purple flowered pea plant with a purebred white flowered pea plant, in his F1 generation, he got 100% purple. Not a blending, no white snuck in there, it was 100% purple. When he allowed those F1s to reproduce and self-pollinate, in the F2 generation, the purples sorry, the purples came back. Not shocking, because purple's dominant. 3 out of 4 times in the experiments that he ran. 25% of the time, the white came back. It didn't matter which experiment he was running. Let's look at axial and terminal. Here, axial is dominant. If he crossed a purebred axial and a purebred terminal, 100% of the F1 came out axial, which is dominant. And when he ran the experiment to get to the F2, 3/4 came back axial, and the recessive terminal came back 25% of the time. So the question is, what's going on here? And so Mendel was able to explain all of this by using letters that he assigned to the problem. So here in my P generation, I'm going to be looking at green and yellow pods. Green is dominant. Remember that if I'm a green pod pea plant, I got an allele from mom's egg and an allele from my dad's sperm. If you're green, both letters have to be capital. And if you're purebred, they both have to be the same. If you cross it with a purebred yellow, that means both parents gave you the same allele, and because it's recessive, we use a lowercase letter. That means that when these organisms right here go to make their sperm and egg, this organism can only pass on a capital allele. This organism can only pass on a lowercase allele. The way we keep up with alleles is by using something called a Punnett square. In the Punnett square, you take the allele combinations for one parent, you take the allele combinations for the other, and you put them on the sides of the Punnett square. You then fill in the boxes. So if this sperm and this egg were to unite, it would make a baby with that combo. If this sperm and this egg unite, you get that. Same here, same here. Interesting. So in the F1 generation, if you want to look at what they all look like, they all 100% looked green. And it's because when you look at their letter combination, every single one of them had a dominant and recessive, but the dominant one. All right. So what's going to happen if we take these and let them self-pollinate? That's like letting them reproduce by themselves or with themselves. So now we make a new punnet for the F2 generation. That's going to give us these results. And in the F2, we see we've got big G, big G, big G, little g, big G, little g, and little g, little g. Interesting. So now we've got 1 big G, to 2 big G, little g's, to 1 little g, little g. And if I want to look at what they look like, this one will be green, so will these. I'll have 3 green to 1 yellow. Oh, here's that 3-to-1 ratio again. Guess what? It's not a coincidence. It doesn't matter which trait you start with. If you were to do purple and white, you could do purple here, white here. All the F1 are going to look purple. And in the F2, the purples will dominate 3 out of 4 times, with the white flowers coming back 25% of the time. So when Mendel kept on getting these statistics right here, he knew math was involved and that blending and haphazard events don't happen during heredity, that there is some math involved, especially for the expected ratios. So now let's start to use some scientific terms. You know what a gene is, right? You know what a locus is. It's an area on genetic pieces. You know at the locus there has to be 2 alleles, one from mom and one from dad. For this chapter, we're going to talk about the fact that most of the time there's a dominant and recessive relationship between the alleles. And when we talk about dominant and recessive, we'll use uppercase and lowercase letters. The way something looks is called its phenotype. I always think of physical appearance and phenotype. Here, we're talking about the color green and yellow. So if you talk about the phenotype of the G1, they were all green. If you talk about the phenotypes of F2, you had 3-to-1 green, yellow. Genotype has to do with these things, the letter combos. That's genotype. So here I have what's referred to as homozygous dominant. Homo meaning same. Here the letters are both capital, meaning homozygous dominant. Here's the complete opposite, homozygous recessive. Here you have both little alleles. These organisms here are referred to as heterozygotes. If homo means same, hetero means other. And here you have one of each other kind of letter. So with that knowledge, now we're ready to work some problems. So if we go to the practice page, let's work number 1 together. Here we're looking at flower position. The alleles are axial and terminal. I don't think you necessarily remember, but axial is dominant. So when you're assigning letters, you have to use the same letter for each allele, but one gets capital, the dominant one, and one gets the lowercase, the recessive one. So now let's read our problem. We've got a homozygous axial. Well, it has at least that allele, and it's telling you it's homozygous. And you've crossed it with a terminal flower pea plant. If it's terminal, it has to have the lowercase allele. But we know it has to have 2 of them, because if you were to slide or give a capital or dominant allele to this lowercase allele, this would actually end up being axial. The only way you can be terminal is to be completely recessive. So make your punnet. And on down the road, when you get really good at this, you may not even have to draw your punnet. You might be able to do it in your head. But here I can see that if I cross these 2 parents, I'm going to get a genotypic ratio. That has to do with the letter combos. I'm going to get 4 heterozygote to 0 of any other combo. I make my zeros with a slash. Don't think that's like a special biology genetics thing. That's just how Miss Keene makes zeros. And for the phenotypic ratio, what will they look like? You will have 4 axial to 0 terminal. Let's work another one. Heterozygous brown-eyed man and a blue-eyed woman. The gene is eye color. We're going to try to keep it simple today by just saying there's 2 alleles, even though we know there's 3. We're going to say only 2. We're going to keep it simple. Brown and blue. Brown is dominant. There are more brown-eyed people in the world than blue-eyed. And so now we need to cross a heterozygous brown-eyed man. So he has brown eyes, but it tells you that he's heterozygous. You have to cross it with a blue-eyed woman. If she's blue-eyed, both of her alleles have to be recessive. Otherwise, if you were to put a capital B, it would overshadow the recessive, and those people would, that woman would still have brown eyes. Only way to be blue-eyed is to be completely recessive. So draw your punnet. Fill it in. Your genotypic ratio is going to be 2 heterozygotes, 2 homozygous recessive, and phenotypically that's 2 brown eyes to 2 blue eyes. So cool, if you're a geneticist, you could tell them you have a 50-50 shot at blue and brown eyes with your babies. Could they have all brown-eyed babies? Yes, because every pregnancy goes back to this punnet. It's still 50-50 for every pregnancy. So even though they have a 2-to-2, 1-to-1, or 50-50 shot at brown and blue eyes, they could have all children with blue eyes. But every single pregnancy goes back to this punnet and was 50-50. Okay, see if you can work the other 2 and then come in and see me if you need help.